Okay, warm welcome to everyone into our platform, that is the Progressive Medical Lab Technology. And today we're gonna discuss about the gram staining. Before understanding the gram staining, we need to understand that the gram staining is the major stain in the microbiology. Into the microbiology, there is a most prominent stain, that is the gram staining. That is basically developed by the Christian Gram. See here, today we're going to discuss why basically we are doing the gram staining, as well as what are the principle of gram staining, as well as we will getting the idea regarding how do we do the gram staining. Into the laboratory, you might have performed lots of time into the gram staining, but you have the lack of knowledge regarding the gram staining. After completing today's class, you will not face any kinds of doubt regarding gram staining. It's our guarantee. See, firstly, we need to understand that why we will do the gram staining: to identification of the bacteria. And whenever we are identify the particular bacteria, is it gram positive or gram negative, it helps to treating the particular patient, that which kinds of drug we have to give to the particular patient to diagnosis it, is it gram positive or gram negative. Okay, to diagnosis or identify the bacteria, we have to do the gram staining. Basically, into the previous class, I have already told you that basically there have two type of bacteria. One is gram positive and another one is the gram negative. Let me explain what are the difference between the gram positive and gram negative. Yesterday I have already discussed the particular thing. See here, that is a gram positive, that is a gram positive, and here is a gram negative. If you are noticing here, the gram positive, that is a peptidoglycan layer is very much thick, and there is a peptidoglycan layer is thin. Layer is thin. Into the gram negative, see here, into the gram positive there is a teichoic acid basically present, and into the gram negative there is a teichoic acid not present. Okay, into the gram positive bacteria there is no LPS, but into the gram negative bacteria there should be present LPS or lipopolysaccharide. Okay, and there have also lots of example of gram positive and gram negative bacteria. We have already understood the particular things into the previous classes. Okay, so that there have the major difference into the gram positive and gram negative bacteria. The gram positive bacteria have the thick peptidoglycan layer, and the gram negative bacteria have the thin peptidoglycan layer. It is the major difference between the gram positive and gram negative bacteria. Okay, now we are understanding that how do we do the gram staining. Before understanding the procedure, we need to understand the principle, how it's happening. See, this bacteria possess the thick layer of peptidoglycan. I have already told you that, see here, that is the peptidoglycan layer is thick into the gram positive bacteria. Okay, so that whenever we are using the crystal violet at first, during the staining, it basically persists into the particular bacteria, so that the purple blue color basically happening, or the bacteria will take the purple blue color. If you are noticing here, see, the gram positive bacteria thick peptidoglycan, so that whenever we are using the crystal violet at first, this crystal violet will take color to the particular peptidoglycan layer or to the cell wall. Okay, so that it's basically purple or blue in color. After that, whenever I am coming the gram negative bacteria, it's also very much important things we need to understand, that into the gram negative bacteria, see here, there is a thinner peptidoglycan layer. Getting my point? So that have the outer lipid membrane, or we can say that lipopolysaccharide basically present. Okay, because their peptidoglycan is thinner, so that they lost the crystal violet dye. Getting my point? But whenever they become extract out the crystal violet dye, they should be counter stained by the red dye, that is the safranin, so that it become color the pink color under the microscope. Getting my point? Within the gram negative bacteria, there should be less or thinner peptidoglycan layer. Please keep in mind that, and there should be, whenever we are using the crystal violet, they will extract out, because we are using the acid also here. Okay, or alcohol we are using here, so that they will extract out, and counter stain should be happening by the safranin. Okay, you will understand the particular things later. See, now we are coming to, sir, that is the how it's happening. See here, that is the procedure. See, firstly take a clean and grace free slide. We have to take a slide. Okay, we have to take a slide. After that, what will you do, sir? After that, we have to prepare the smear of the particular specimen, okay, or the particular bacterial specimen. We have to do the smear, or we have to take the smear. Okay, we have to perform the smear. After that, what will we do? After that, we have to edit the crystal violet. Okay, whenever they become dry, we have to add the crystal violet. Okay, and the crystal violet whenever we are adding, so that there should be two types of bacteria. One is gram positive, another one is the gram negative. The gram positive bacteria takes the crystal violet, that is a pink in color. Okay, that is a pink in color, and that is a see, that is a pink, not pink to violet in color. Okay, that is a violet in color, crystal violet. So that blue to violet in color. See here, purple to blue in color. Okay, it will take. Okay, after that we have to add it 30 seconds to 1 minute. Okay, that basically depend upon the website to website, as well as book to book, as well as into the laboratory to laboratory. Okay, there might be minor change you might see into the laboratory based. Okay, after that, what will we do, sir? We have to added the grams iodine. Grams iodine should be added at the mordant. Please keep in mind that crystal violet are acts on the, I have to choose them, take the another pen set, that is the crystal violet basically are doing act into the primary stain, getting my point, and grams iodine basically act on the mordant. And what do you mean by mordant, sir? Mordant basically help to create the environment so that the stain can attach the particular surface properly. That is the function of mordant. Okay, after that 1 minute, after that we have to wash with the water. Okay, so that whenever it will be washed, the gram positive bacteria should remain into the violet color, okay, and gram negative bacteria is also right now violet in color, because that is a crystal violet I have taken. After that, whenever we have to use the 95% alcohol or ethanol for 10 to 20 seconds, because whenever we are increasing the time, there might be chances to extract out everything. So that we have to keep in mind that. Okay, now we are just giving the acetone or ethyl 95% alcohol. See what will be happening? They should remain violet, but their color should be extract out, so that I have to erase the color. The bacteria is remain same, that is a gram negative bacteria, but the crystal violet should be extract out. Okay, so that the bacteria is now colorless. Okay, but this bacteria is violet in color, gram positive bacteria, and gram negative bacteria is completely colorless. Okay, after that we are adding the safranin. What do you mean by safranin, sir? Safranin basically added for the counter stain. Okay, we are getting that primary stain, that is a crystal violet. We are adding that mordant, that is a grams iodine. We are adding that is a counter stain, that is the safranin. Okay, and whenever we are adding the safranin, there should not be any kinds of impact, so that they are basically counter stain are happening. Okay, the background become stain, but into the gram negative bacteria, they basically takes this counter stain. How it look like, sir? That should be looking like a pink. I have to take a pink pen. There is a nothing pink pen, so that I am just defining here, that is a pink. Okay, pink, so that the gram negative bacteria basically taking the safranin's color, and the gram positive bacteria basically taking the crystal violet color. Getting my point? So that it is the basic difference. Okay, so I hope the principle is already clear. If any kinds of doubt, just write down into the comment box. Okay, and after completing the particular things, we have to wash the slide properly and go for the microscopy. Okay, see, I have to erase the particular things. See, go for the microscopy. After seeing the microscope, if it's violet in color, it defining the gram positive. Okay, and if it's pink in color, it defining the gram negative bacteria. Okay, so that you should not be forgot the particular things. Okay, gram positive and gram negative bacteria we have already understood. See, now here is a two types of bacteria that is mentioned. That is a gram positive bacteria, that is a blue to violet in color. That is a gram negative bacteria, that is a pink in color. Okay, after seeing the microscope, we can easily detect the particular things. Now here is some kinds of bacteria. They basically gram positive bacteria and gram negative bacteria. It is a very askable question in every kinds of examination, that for the beginners. See, there is a gram positive bacteria: Staphylococcus aureus, Streptococcus pneumoniae, Bacillus anthrax, Clostridium tetani, Listeria, Enterococcus, Mycobacterium tuberculosis. Okay, Mycobacterium tuberculosis is the controversial thing. Sometimes it's gram positive, sometimes it's gram negative. So that just erase the Mycobacterium tuberculosis. Okay, after the Corynebacterium diphtheria and the gram negative, that is the Escherichia coli, Salmonella typhi, Klebsiella, Nigeria, Shigella, Vibrio cholerae. Okay, all enterobacteria are the gram negative bacteria. Okay, and that is the basic difference between the gram positive and gram negative bacteria. I have already told you that the gram positive bacteria is the thick peptidoglycan layer, and the gram negative bacteria, that should be the thin peptidoglycan layer. Gram positive bacteria basically looking like a purple in color, and gram negative bacteria, that is a pink in color. Into the outer membrane, see here, that is into the gram positive bacteria, that is absent, and gram negative bacteria it's present, or LPS, lipopolysaccharide. Okay, see, the teichoic acid should be present into the gram positive bacteria, and into the gram negative bacteria, that should be absent. And LPS, I have already told you that into the gram negative bacteria, that is gram positive bacteria, they should be absent, and whenever we are coming to the gram positive bacteria, that is present. Okay, got it? I think that the particular thing is clear. Example, I have already told you that. Okay, thank you so much for listening. Thank you so much for attending. For further next class, just give a thumbs up and share with your friends, and subscribe our YouTube channel, and just check the description for all kinds of lab technology updated. We have the WhatsApp group, we have the Telegram channel, Facebook, as well as every kinds of platform we are available. Just check the description box and share it in the maximum level for the further classes. Okay, thank you so much for listening.